Welcome, everyone. I'm sorry about the time. We couldn't get this auditorium any other time, and Susan couldn't make other times, so we had to arrange it this time. And I know it's after term was finished and everything, so it was a bit of an odd time, but you've got an audience, Susan. It's fantastic. Okay, so before I start, I need to acknowledge the Eva Kavan Prize winners. These are for the graduate students for excellence in research. And the committee decided there was going to be 2 winners this time. It was pretty for the first time. They couldn't decide between them. And one of the winners is Anne Collins. So Anne is right here. Anne works with Kate Wassam and explores the cholinergic interneuron activity. I think we've got a nice picture of her here. And uses lots of biosensors and optogenetics and all these nice new techniques to study the role of, how did that happen? Oops, we've got an automated thing going on here. And looks at the role of these interneurons in the striatum, specifically on some reward-related behaviors. So, Anne, I've got a little prize for you here, so congratulations. So the second student is Hua Chaya. Is she here? Come on, you guys can't sit down there forever. And hers a, did I say your name right? What? Is this a Wa? I always thought Wa, it was HOA, but never mind. It's lots of different spellings of everything, yeah. She's an MD Ph.D. student. She's been working on astrocytes. And actually, she's been working with Anna a little bit in my lab, so that's really cool. Her PhD projects showed that astrocytes from the striatum and the hippocampus are functionally, morphologically, and molecularly distinct. And she uses lots of different techniques, electrophysiology, calcium imaging, EM, and transcriptomic and proteomic analysis. I'm sure she's going to do great. Having an MD PhD is really a ticket these days to get on with your lives. So congratulations, Wa. Okay, so next is introducing our Magoun lecture this year. And I personally like to congratulate Susan on this honor. She was chosen by a committee chaired by Baljeet, and in my view, it's a completely outstanding choice. What strikes me about Susan, actually, is her breadth. Whenever I talk to Susan about anything, she always has something to say about it. Anything from, aging, which we're all doing, and autism, and even drugs of abuse these days. I think she's been getting into, especially looking at some of the cannabinoid stories. She works with imaging mostly, and I think her favorite brain area must be the hippocampus, because she is nodding, because she's always talking about the hippocampus. She's a clinical psychologist and really studies, as I said, many different areas of neuroscience. She received a BA from Cornell and then a PhD from Wayne State. And then following an internship and 2 years as a lecturer at Yale, she moved to NIH for a postdoctoral fellowship in epilepsy. And she was recruited to UCLA in 1993 and has lots of collaborations now and helped develop the functional MRI program here. So Susan holds the Joaquin Fuster chair, and Joaquin is right here, so that's good. She's a professor in the Department of Psychiatry and Biobehavioral Sciences. She leads many, many different scientific efforts at UCLA. She was named director of the Staglin Center for Cognitive Neuroscience in 2012, and that houses all of our MRI laboratory and EEG studies for the Semel Institute, and others, I guess. In 2015, she became director of the IDDRC, and she's PI of the New U50 Center Grant, which was awarded in 2015. She's been PI of the UCLA Autism Center for Excellence Grant for the past 10 years, and that was just renewed for another 5 years, so congratulations on that. And she's also the PI of several large-scale multi-site initiatives, including the Human Connectome Aging and Human Connectome Development projects, as well as the Autism Girls Network. What's that? I don't know. Okay, so I think I'm really happy that Susan was chosen. She's published many different papers. She has one of the highest H indexes of the scientists I know at UCLA, and I'd like to congratulate her on getting this Magoun lectureship. Congratulations, Susan. And she has a nice little memorial to say she got the award.
Great, thank you. Thank you, Chris. I really appreciate it. This is a tremendous honor for me. Great getting it from Chris Evans, who's just been such a great leader of the BRI and really appreciates, I think, breadth and different kinds of research. I note that I'm the first brain imager to get this honor, and which is interesting because UCLA has been at the forefront of brain imaging since the early 1980s. I'm also the 5th woman, I believe, to get this award, so I hope that that is marking a new trend towards more diversity. And today I'm going to talk about the hippocampus, and it is in fact my very favorite structure because it's so pretty, and I'll show you pretty pictures as well, from an imaging perspective. And I hope to convince you that, that research in neuroscience can be done on the human being. So I will talk more about that. But first, I would like to thank a few people, and foremost is Dr. Joaquin Fuster, who endowed my chair. He's been a great mentor and a role model for me. I'm so grateful for him for so many things. And I'd also like to thank my department chair, Peter Wybrow, who's just been amazingly supportive of me and a great leader, and the reason why I'm still at UCLA. I'd also like to call out my close colleague, Allison Berggren. I've been working with Allison for a very long time. She did her PhD with me and her postdoc, and she's now an associate professor, so we've been together a very long time. And she's been spearheading some of the hippocampal high-resolution imaging studies that I'll be talking about today. And finally, my colleague, Mirella DiPreto. We do autism research together, so nothing I'm going to be speaking about has anything to do with Mirella's work, but she's been such a close colleague. We've been together longer than either of our marriages, so, so I have to call her out. So I wanted to just start with a case study, and this is a famous case study, but I was interested in it because one of the very first patients I ever saw when I was doing my internship at Yale was of a young man, a 21-year-old, who had come to the emergency room with a high fever, a rash, and a little delirium. And back in the early 80s when this happened, we didn't know very much about herpes encephalitis, but at Yale they were pretty sharp. The resident immediately recognized it for what it was and shot him up with a new drug called acyclovir, which saved his life, which you may think is a blessing or a curse, because from then on he was not able to remember things for more than about 30 seconds and expressed a shock every time he saw someone new about where he was and what was going on. And it's very much like this more famous case of Clive Waring. And if you haven't seen the videos on Clive Waring, I really strongly recommend that you take a look at them. There are a bunch of them on YouTube. It's very moving and very, just very illuminating. So this is Clive Waring. He was a musician, pianist, and conductor who experienced herpes encephalitis and is now living in a home with very, very severe memory deficit. But what's interesting, and what I hope to base this talk on, is the nature of his deficits and how very human his memory deficit seems, how much it resonates with what we think about when we think about humanity. So this is his wife, Deborah, and he has never forgotten his wife, Deborah. They don't live together, but she comes to visit him, and whenever she comes in, he expresses this amazing surprise, and he's overjoyed that she's there. And in fact, when she leaves the room and comes back in, he greets her with that same incredible surprise and joy, which is kind of scary, but touching in a way. But what's interesting is that he's been sick for many years now, almost 30 years. And so this is his wife as he knew her early on, with a lovely big hair of the 80s. You will all recognize the big hair. And as a redhead, and then later on as a cropped-haired blonde, as a curly-haired blonde, and this is what she looks like today. Interestingly, he has to remember stuff because he remembers his wife, and when he sees her, he's delighted to see her, and he doesn't say, "What have you done with your hair?" or "My God, you've gotten fat," or anything like that. He recognized her, so he's clearly has, in fact, remembered her. But what is missing is this idea that he knows her, this ability to say in a conscious way, "I am aware of having seen you today and previously." It's that part that's missing, this ability to declare, "Yes, I have learned this," as opposed to the learning itself. And this is his diary, so he keeps a meticulous diary. And he writes in it pretty much all the time, and you can see the timestamp, and there are books and books and books of this. But at every moment, he suddenly feels, "Oh my gosh, right now I'm conscious. I'm awake for the very first time. I am now truly, completely, totally aware." And then 20 minutes later, he writes that, "Now I am superlative, completely awake." And he crosses out what he just wrote down a few minutes ago because he wasn't awake then, but he is now. And all of this goes on and on for his entire life. So at any one moment, he's aware of who he is at that moment, but there is no continuity of consciousness. And so this is how we describe the memory deficit, as this essentially a deficit in consciousness. He's lost in time. And similarly, we know, of course, a lot about our famous case, HM. I'm not going to go into any detail here, but we have a picture of his brain, so I thought I'd show you his brain. Here's his brain with his hippocampus missing. Here is the brain of someone else, and that's what is still there. And this was an epilepsy patient. I started my career in epilepsy, and one of the things that we have to do is map memory using, in the old days, exclusively the WADA test, and hopefully in the future, functional MRI. But one of the things that was interesting about HM was that he did learn. He was able to learn. He could learn to navigate his environment successfully, even though he was in a new environment. He was able to learn skills such as mirror reading, reading backwards, which is actually pretty hard to do. And he was also able to learn some complex problem-solving tasks like the Tower of Hanoi, which the first time you see it takes a very long time to learn, but you can figure it out after a few trials. The minimum number of trials you can solve the task is 31, and he can solve this task in 31 trials, but he's never seen it before in his awareness. So there's this disconnection between what you're aware of knowing and what you actually know. Also in epilepsy, we care a lot about content specificity, language versus visual spatial knowledge. So when we talk about then the hippocampal memory deficit in humans, we refer to terms that seem very uniquely human. We talk about this loss of the continuity of consciousness, that they're lost in time. We talk about self-awareness, conscious recollection, reference to specific episodes. In fact, we call this declarative memory. I can declare, which presumes a language component. And indeed, when we do our pre-surgical mapping, we are looking for lateralization of language function so that we don't disturb verbal memory. These aspects of human amnesia seem tied to what I think we would all agree would be very uniquely human abilities. And in a way, you can think that's very surprising, since the hippocampus is quite an old structure, and it's very similar to that of other animals such as rodents. In rodents, we talk about other things. We talk about like novelty detection, navigation, including cell-specific functions like place cells, grid cells, time cells, and some associative learning, associating, for example, food with a specific location, and also fear acquisition. Not a lot of similarity between how we would describe the human amnestic experience with the rodent amnestic experience. So the questions that I'm going to try to address today are, how can we reconcile the uniquely human characteristics of episodic memory that we talk about when we talk about the hippocampus in humans with what we know about the hippocampus in lower animals? And I'm going to do this by using some high-resolution functional MRI techniques, which I'll tell you about, of the human hippocampus. And at the end, I will mention some single-unit recording studies as well in human hippocampus in comparison to animals. The second question I hope to touch on at the end is this one that's always mystified me. If the hippocampus is so very critical to what makes us so uniquely human, this aspect of being able to follow our conscious selves over time, then why is it that the hippocampus is so incredibly vulnerable to so many things that happen in our environment? Encephalitis, anoxia, stress, seizures, and of course, aging. Why don't we just develop visual deficits when we develop Alzheimer's disease? Why does it have to be the hippocampus of all things? And I'm going to suggest that these questions are not entirely unrelated, and I'm going to use the example of the Alzheimer's risk gene ApoE4 to illustrate that. So first, just a real quick look at the hippocampus across species. So we've got rodent, monkey, and human. And I'm not going to go into details here except to show that all species have a nice hippocampus. They have the entorhinal cortex that's attached to it. It's only in a different location in the rat because their heads are in a different location. And if you look at the internal structures, they have a central dentate gyrus, and they've got these CA fields 1, 2, and 3. And they're pretty much oriented in the same way if you account for the difference in the just general shape of the brain. So the internal structure and it looks quite similar. And we also know a fair amount about the connectivity of the structure. This is a simplified model based on mostly tracer studies in monkeys, rats, and some in cat. And I'm sure this is a diagram that you've all seen, but the basic idea is that information comes from many parts of the cortex. It takes a pit stop in the entorhinal cortex before going into the hippocampus, and then can take one of these routes here in the dentate and CA3, where they have these recurrent collaterals, these what we would call reverberating circuits, and then this other pathway through the CA1 and subiculum and back to the entorhinal cortex. And most of this is learned either through tracer studies in slice prep and so forth, and also in single unit recordings. And of course, the problem we have in human studies is that we can't normally do dissections of human brain, and we can't put electrodes in them very commonly and look at function at the same time. We can do some postmortem studies, and this is a study from one of my former graduate students, Michael Zaney, who was in our MSTP program here. And this is a very, very beautiful study where he did diffusion tensor imaging in postmortem hippocampus and also did some track tracing. And I'd like to show this just because I think the picture is so very pretty. It's really a gorgeous picture. But also, I think the main point that he made, and I urge you to look at this fantastic article, it's very comprehensive, is that it looks as if the same kind of connectivity that we see in those animal models we see in the humans. It looks very, very much the same. So same hippocampus, different functions. How do we account for that? The technique that we've been using is functional MRI, and in particular a high-resolution MRI, to look at the hippocampus in humans in vivo. Interestingly, for many years, imaging studies did not show any task-related activity in the hippocampus, and this worried us early on because we were seeing task-related activity in language and motor and every other function we could think of, but not in memory. And there are a number of reasons that were posited for that. One had to do with, for example, neurovascular coupling. In imaging, we rely on neurovascular coupling, that is the relationship between neural activity and the vascular consequences, increases in blood flow associated with that neural activity. And so one idea was that perhaps it's not preserved in hippocampus. There is very high baseline activity in hippocampus. The hippocampus is working all the time because it's constantly encoding what our experiences are. And so maybe there's just no room for change. Maybe it's just a change in pattern. There's no change in magnitude. Another possibility is that the tasks or the control tasks could have been very badly chosen. And there's some really prime examples of that. There was an example of an early PET study that was published in Science. And the task was to memorize a list of words. And the control task was to read a list of words, but to not try to memorize it. So that's like me saying to you, okay, I'm going to tell you some animals. Remember camel, but don't remember yak. Okay, so everybody remember camel and don't remember yak. Let's see how we do at the end of today. It was absurd. And now we know a lot more about how to design control tasks, and it turns out to be quite difficult. But a third reason, and one of the ones that we decided to tackle, was that, that there are technical reasons for lack of hippocampal signal. First of all, the anatomy of the hippocampus, as we've shown you before, suggests a great deal of functional heterogeneity within these different substructures. And the standard spatial resolution of MRI, which in fMRI has usually been 3 millimeter cubic voxels and 1 millimeter of voxels for structural imaging, it's not really consistent with this heterogeneity, particularly when the hippocampus is rolled up as such. So if I look, for example, at these are 3 millimeter voxels, you can see that a voxel here might include some part of the hippocampus, but it also includes some other region. I think this is the subiculum right here, and you could include some CSF, and you could include some white matter. So it might be very hard to get a signal in these voxels, these 3 millimeter voxels, that will give you a homogeneous signal of a part of the hippocampus that's all doing the same thing. So it's a pretty serious problem. And it's exacerbated by, of course, this rolled structure of the hippocampus, which, as you know, has been described as a jelly roll. Now, nobody really eats jelly rolls anymore, right? These are repulsive. It's like a Twinkie. It's like a Twinkie with spuckers in the inside. It's really gross. But it's important to understand the hippocampus, to understand the structure, so I like to redefine this as hippocampus as Cinnabon. Cinnabons are so much nicer than jelly rolls. And they've got this beautiful cinnamon layer and a lot of brown sugar layer, and they've got the icing layer and, of course, the nice dough. And if you're lucky, they've got a caramel and a pecan layer in the inside as well, all rolled up. So if we wanted to visualize all of these different layers, we would need higher spatial resolution to look at those substructures. And it would also be really nice to unroll this into a single sheet so that we could see functional patterns across the whole structure more readily. And of course, the problem that we have is that when we start to go to smaller and smaller voxels in MRI, we lose signal as we go. So there's always a trade-off between voxel size and SNR. So how can we deal with that? Well, so if we think of the Cinnabon along its long axis, in the same way we would think of the hippocampus along its long axis, so here's the sagittal view and this is the cross-section, you can see that a lot of that heterogeneity of internal structure is in the cross-section here. But along the long axis, there's not much heterogeneity. So for example, here we've got the dough layer, the external dough layer all the way across the Cinnabon. And we've got the sort of the internal brown sugar layer all that goes from front to back, and the central core of the gooey center right in there. And then it also goes front to back. And the same thing is true in the hippocampus. So what we can do then is use anisotropic voxels. So voxels where we have very, very small voxels within the cross-section where all of the heterogeneity is, and then which would normally lose a signal, but then we can regain signal by making the voxels long where there is little change across the long axis. So there is some change, obviously, across the long axis, particularly when you get into the head, but certainly not at the resolution that it is in the inside of the hippocampus. So by using these long voxels, we recover signal while maintaining functional homogeneity within the voxel. So this was work that was originally done in collaboration with my former graduate student, Michael Zaney, and there he is again. And he was a very good programmer and helped us develop an approach for segmenting and unfolding the hippocampal complex. And so I'm going to go through the procedures and then I'll start looking at the results. So this uses, for those of you who are MRI geeks, a T2 fast spin echo pulse sequence, which has very high gray-white contrast and also has the advantage of not having a lot of susceptibility artifact in the base of the temporal lobe, which is a serious problem for imaging when you're imaging the base of the temporal lobe, which is what we're doing. It's a small field of view, so basically we just cover the hippocampus, but we cover it in these slices that are perpendicular to the long axis of the hippocampus, giving us the maximum resolution within along this long axis and within those voxels in the cross section. So in the old days, we had voxel size that was structurally 0.39 voxels by 3 millimeters. We now do it at 2 millimeters and slightly less than 0.39, which is substantially better than how we had it before. And the functional voxels are about 1.5 millimeters. Still large, but much smaller, and making it possible for us to look more closely. So then the technique is to first look in the cross-section, identify these different subregions, and we do this just with reference to an atlas. Some of them we really can't distinguish. So for example, we don't have the resolution here at 3 Tesla to distinguish between the central dentate and the CA3 and the nearby CA2. So we put it all in one region that we call here, but we can get the CA1, the subiculum, parahippocampus posteriorly, entorhinal cortex anteriorly, and so forth. And so what we do then is we segment the image by first identifying CSF, then identifying the white matter, and then growing a gray matter ribbon right along here. So that has all the gray matter throughout the whole complex here. And we do this for every slice. You can extract that and then form a sheath. So this is now a 3-dimensional sheath of the hippocampus anterior to posterior, medial to lateral. And then you can imagine just taking this sheath and pulling it out and flattening it. And that's exactly what we did, creating this, which is a flat map. And because we have these boundaries here that are created on individual slices, we can maintain those boundaries as we flatten out the hippocampus and thus have all of these lines which separate the different subregions of the hippocampus in flat space. And through the magic of image processing, we can bring different people's hippocampi together, take these boundaries, normalize against the boundaries, and then just do any stretching and pairing that we need to do within the region, maintaining the boundaries so that when we do these measurements, my CA1 is the same as your CA1, my CA3 is the same as your CA3, and so forth. So that's the basic technique that we're using. So what I'm going to be talking about are patterns of hippocampal activation across different memory tasks and asking whether we can examine and come up with some basic principles for a functional anatomy of the human hippocampus and determine ultimately if indeed this conscious recollection that is so important for our memory impaired patients is indeed a necessary part of hippocampal activity in humans. And then we'll compare that to some tasks that are used more widely in rodents. So here's the simplest of tasks. This is a novelty encoding task, and there are many comparable novelty encoding tasks in rodents. Of course, they look at different aspects of novelty, usually smells. Here we look at scenes. So subjects see a series of scenes like this, and all they have to do is to look at the pictures and say whether these scenes are indoor scenes or outdoor scenes. That's just for us to make sure that they're paying attention to what they're doing. And then we have a series of blocks of all new pictures each time, or the same 2 pictures repeated again and again and again. And we do this over time. So novel versus repeat, and that's the comparison that we do. A very simple block design. These are the basic methods. We use a very long TR for those of you who are MRI geeks like myself. We use a longer TR that just gives us more signal, which is very nice. We also do this thing called we normalize the activity in each block against its neighboring blocks, and that's to account for signal drift, which used to occur quite a bit in the old MRI scanners, not so much anymore. And so very simple contrast of the new versus old blocks, but now that we can do this in flat space, we can extract the time series from each individual subregion of the hippocampus. So let's see what that looks like. First, let's do it visually. So these are flattened hippocampi. This is the right hippocampus. This is the left hippocampus. And these are the demarcations of the different subregions, and this is the key to it. But the first thing I want you to see as you look at this is that it's not the case that we see activity sort of evenly spread across the whole hippocampal complex. In fact, it's quite restricted. And it's restricted to a couple of areas right here in this anterior CA3 dentate gyrus region, and that's bilateral. We also have some activity in the spiculum. Notice that there's essentially nothing going on in the CA1 or in the entorhinal cortex and perirhinal cortex in this study. And then we also see this bilateral activity over here in the parahippocampal gyrus and in the medial part of the fusiform gyrus. And this is activity that we know to be associated with the parahippocampal place area. So that's scene processing, a complex visual task, visual processing, visual association cortex in humans, and this is an object recognition center. Both are nicely bilaterally organized. So there they are, like that. And you can see how nicely comparable the left and right hemispheres are. Now we can look at the pattern of activity within those subregions and ask ourselves, are these subregions functionally distinct? And so I divided up these time series curves. So the gray is the task, the white is the repeated ones, and we can see a couple of different patterns. So first, for example, in the parahippocampal cortex and the fusiform, which we believe are visual association areas, we see a simple effect where every time they see new things, there's more activity there, and then compared to those new blocks, there is less activity when they're seeing the same items over and over again. So very straightforward. I'm going to skip over to the CA1 and the entorhinal cortex, where we see no activation whatsoever associated with a task. What you can see if you look at entorhinal is a lot of spiking up and down, so a lot of variability in activity here, but not task related. We'll get back to that later. But of interest is this emerging activity over time in both the CA3 dentate and in the subiculum, where we start out with very little differentiation between the all different items and the items that are going to be repeated again and again. And over time, we see a greater differentiation, which we believe is just associated with the learning. That is, the learning of those repeated things. They're no longer adding new information. There's less activity there in comparison to where they're still seeing new information. So we see a learning effect. We see sort of a sustained visual processing effect. And then in these regions, we see nothing at all. So evidence for heterogeneity in the function across these hippocampal subregions. So what about a task that's a little bit more important to humans, particularly those of us as we get older, find this to be one of our largest problems, associating names with faces and trying to remember those associations. And these are, of course, difficult because it's a random association, right? There's nothing about this face that says it should be named Diane. It's just purely random, and we have to bind these two ideas together. And so we did this task with multiple blocks of learning, recall. So here they learn the association. They'd have a little distractor, which was just to press this button every time a crosshair came up. And then they'd see the face, and they'd have to generate the name. And they do this over time, same groups of faces, so we can look at learning curves and learning effects. I'm going to show you the same hippocampal flat maps. It's a little more complicated now because here we've got left, and right, but we also have the learning blocks and the retrieval blocks. And what I hope that you could notice right off the bat is that this area in the anterior hippocampus associated with learning is very specific to the encoding blocks, not found at all in the retrieval blocks and the recall blocks, even though they're interspersed. So the activity is going on and off and on and off, depending on whether they're encoding or retrieving. They're also bilateral, and you might also notice that this is very much the same region that I just showed you in the previous task. You will also see some differences out here. So now instead of having activity in the parahippocampal region and in the medial fusiform, there's activity in the lateral fusiform. And it's greater in the right than in the left, and that makes sense because this is where the face processing areas are. And the face processing, although there is some bilateral representation, it's much greater in the right hemisphere. So again, we believe that these are material-specific areas, but these are not. We look at the time activity curves again, we can still see these different patterns, and we can see something else. During the encoding blocks, in this anterior CA3 dentate gyrus region, we see activity when subjects are first seeing all those new faces. But as they have come to learn them, and these are young, healthy volunteers, so by the third trial, pretty much most of the students have learned all of these associations, the activity goes down as the information has been successfully encoded. And similarly, we see the same thing during retrieval, but again, we see activity during retrieval primarily in the posterior subiculum. We see it for both encoding and retrieval, although it's greater during retrieval. In contrast, in the fusiform gyrus, we see a lot of activity the very first time they see the faces, and then after that, it's pretty much even all the way across. And it doesn't matter whether they're encoding or retrieving those faces. And so what we're seeing then is that there's an anterior, posterior, and regional dissociation between learning and recall, where encoding is primarily in this anterior CA3 dentate region, retrieval is primarily in the posterior subiculum, and then this really is not memory related so much as it is visual processing related and priming related. So now I told you before that we couldn't quite distinguish between the CA3 and the dentate gyrus because we didn't have the resolution at 3T to do that. But at 7-Tesla, you can do that. So my former graduate student and now assistant professor here at Nathya Sutana, who's truly amazing, by the way, went off to our colleague Bob Turner's group in Leipzig, Germany, where they had a nice 7T magnet. And they were able to reduce the fMRI's voxel size and get really, really good spatial resolution and a good signal from a 7 Tesla magnet. And they used a very similar face name encoding and retrieval paradigm, except instead of having names like Robert and Henry, they were named like Gunther and Helmut. They were in Germany. So this is the same MRI, but at 7T, and you can see that you can really see the subregions very, very nicely in humans, and you can segment them very well and distinguish between the central dentate and these other parts of the CA fields. And of course, the flat maps look a little different because they're more complicated because we can see more different subregions. This is a direct comparison between learning and recall. So the blue are the recall areas, the red are the learning areas. And I think that you can still make out that you have this anterior-posterior dissociation. We also have this, you can see down here, if you compare the dentate and the CA3, that actually most of that learning-related activity is in fact in the CA3 region. There is some in the dentate, but it's mostly in CA3. And again, we see that same dissociation between So how about something that looks a little bit more like something that rats like to do, which is navigate? So Nanthia did this other study looking at spatial navigation. This is a task where subjects were just brought to different locations using different kinds of routes and brought to these landmarks. And over time, they would learn this fake city that they were being brought around in. So in encoding, subjects viewed navigation to novel stores, and then during retrieval, they viewed navigation to previously learn a target or else novel or lure star locations. Again, this is a direct comparison between encoding and retrieval. And here what you see is the exact same dissociation between the anterior CA3 dentate gyrus region and this posterior subiculum region. This time it's restricted to the right hemisphere. So some evidence for material specificity by hemisphere, and nowhere else in the hippocampal complex, which is kind of interesting. And if we look across tasks, what we can see is a great deal of consistency. So even though these tasks themselves are very, very different, we see the same regions in the hippocampus that are lighting up. And both for encoding and retrieval, and where we see the differences, of course, are in the material specific stuff, but that is outside the hippocampus proper. And so if I'll first just generally conclude that it does seem like there's a great deal of process consistency in the studies that we've shown so far with this encoding retrieval dissociation in different regions. But let's go to the question that I brought up earlier, which is to what extent does this have to do with conscious recollection of episodes in humans? And so this has been studied behaviorally and with imaging using this task called the remembering versus knowing task. So remembering refers to our ability to, in a sense, re-experience the encoding event at the time of recall. It's a true episodic memory. So for example, I can remember the very day that I first met Ann Andrews, and I can describe that particular event. That's a good episodic conscious memory I have of her. But sometimes we have a different experience of knowing. We walk down the street, we see a face, we know that face, we know that we know that face, but we have no idea where it came from. We really can't refer back to that explicit memory and say, ah, yes, that's where that person came from. So this is a little bit closer to this idea of conscious recollection, and a former student of Barbara Knowlton's and mine, Laurel Eldridge, did this study using high-resolution fMRI during encoding and retrieval of object-object or object-word pairs in an event-related design. I'm going to show you the design. So here's the design. They are shown these word pairs like dinosaur, bread, and a picture that goes with it. So like here, the dinosaur is sitting on top of the bread, and it's a red dinosaur. And then for the retrieval task, which is what I'm going to talk about, they're shown a word like dinosaur, and they're asked if they recognize this word or not, yes or no. And then if they say yes, then they're asked to make a distinction. Do you really remember the encoding episode, or do you just have a feeling that you'd seen this before? And subjects can be trained to do this, and they can actually be quite reliable at doing this. And so the way we do this then is we present a bunch of these different items. So these would be presented items, and the green ones would be foils. But we also are measuring their behavioral response. So their behavioral responses are complex. There could be a lot of different things. For example, this one may turn out to be that they remember seeing that item, but this one might be, well, I know that I saw it, but I just have a feeling. I can't actually remember seeing it. I just have a feeling about it. And then some could be they've just forgotten them. And of course, these are foils. These are correct rejections. And so we have all these different items that we don't know until after they do their behavior, which is what. So what we have to do then is we have to go and collect all of the data from the different trial types. So we know something about the hemodynamic response function. That is, we see an item, and then the blood flow slowly starts to change. It slowly increases, it slowly decreases. We know the timing of that well. So we can model each of these different kinds of responses and then pull them together and then get an average response for the different trial types. So that's how this analysis is done. This is a little bit of a complex slide, but I just want to show you. This is for retrieval. These are the 4 different trial types that I told you about. And I want to point out that the explicit memories are here in red. And this is the subiculum. And again, this is a retrieval task. So that's not too surprising. We also see it in the CA1. Here's the point. The point is that subjects, when they have a conscious declarative memory of the event, then they get this nice activity in these retrieval areas that we know about in the subiculum. But if they truly remember an item that they really did see, but only have a feeling of familiarity with it, they don't have a true episodic memory, they don't know the specific learning circumstances, their activity is just as if they had never seen the item before. It's no different than things that they completely forgot or things that were never there in these retrieval areas. In contrast, the items, if we look back in the CA3 dentate region, which has been associated with encoding, we see activity there for the new items, right? The ones that they never did see before, the correct rejection. So again, supporting this encoding retrieval dissociation, but also showing that it's not just remembering information, it's remembering it consciously. It's by having that declarative memory in humans. So thus far, I've shown you that there is good evidence for subregional functional heterogeneity within the hippocampus that fMRI can show us, and it's consistent across tasks. The same subregions are active across novelty, association, spatial navigation, and this conscious recall. And there's a clear encoding and retrieval dissociation in these different regions within the hippocampus. We believe that this is a similar anatomy to tasks to the extent that they are similar that are traditionally studied in animals. I'll talk a little bit about that. But I think what's important in the human is that it does appear that conscious recollection is important, at least during retrieval in humans. And I don't think that we can ask rodents about that. Now, one question that you might ask is, well, are we really able to ask the same questions about animals and humans? Because really, we're doing functional imaging in humans, but in animals, we usually just stick an electrode in their brain and do single unit recordings. So maybe it's just a single method difference. And what is the relationship anyway between single unit recordings and blood flow changes that we see in humans? So this is something that my former postdoc that I shared with Itzhak Fried, Arne Ekstrom, did. And he used these very cool Benke-Fried electrodes in patients with epilepsy who are being recorded with these depth electrode cordings that stuck into the hippocampus to monitor seizure activity in preparation for epilepsy surgery. So it's a rare opportunity to look at single unit recordings in hippocampus. And he asked the question about what is the relationship between blood flow activity, local field potentials versus single unit recordings. And so the method is relatively simple. These are the electrodes. This is the CAT scan of an electrode. And the reason that we have a CAT scan is because you do not want to run an MRI scan when you put an electrode in somebody's head. That would be really bad. But what we can do is get the CT scan, look at the different electrode points, register it with an MRI scan, figure out where they ended up, and then you put that MRI scan into flat map space, keeping the locations of those electrodes. And then we can find out exactly where those electrodes ended up in our flat map space, looking at the subregions. He did this with a spatial location task, a little closer to what you might do in a rodent. That is, he just had people using a joystick to navigate through a fake spatial environment. And they'd pick up people like that who wanted to, they were like a cab driver. They'd pick up these people who wanted to take them to go someplace. They'd navigate around till they found their destination, like Burger City here. And so this is what he found. So here is the functional MRI that was taken separately. These are the electrode locations on the same map. They extract the time series from the electrode locations so that we can therefore compare the fMRI activity to the single unit and local field activity from those same electrode pairs. And these are the results, and it's pretty straightforward. This is the bold activity. This is the local field potential, and you can see they're very nicely correlated. This is just for a single electrode, by the way. And this is the single unit activity. And there is really no relationship between the single unit activity and either the LFPs or the bolds. This is probably because the coding is very sparse in the hippocampus. We know that, and a single unit activity is just not going to allow the kind of effect that would be big enough to measure. So these two we know are related. And just for those of you who care about this, it's primarily correlating with local field potentials in the theta band, which is, I think, is pretty cool. Now, I mentioned place cells, which is like one of the big things that we do well, I don't do. Other people do it. Rodent cyberodontophobia, actually. And this is a standard place map. So this is a heat map of how many times neurons fired when the animal who was navigating all around this environment showed up in this particular spot. So one question you might ask is, well, okay, this is something that, like, this is the big finding in animals. Do we see this in humans? And using single unit recordings, Arne Ekstrom and his colleagues, Mike Hanna and Isaac Fried, have shown, in fact, that humans also have place cells, and they look just like the place cells, and they act just like place cells in animals. And the same has been later found in grid cells and other kinds of things that have been found in animals. So are there kind of uniquely human place cell equivalents in other content domains? Because, like, place cells might be important in humans, but the truth is that we don't spend a lot of time worrying about that. But we do worry about things like faces. So Nanthia has been doing some really great work looking at faces with Isaac Fried with single unit recordings. And this is an example of just one study, and I'm just going to go over it real briefly. But what they do is they show these same patients with epilepsy hundreds and hundreds and hundreds of pictures of people until they find an electrode that responds to a person. And then they can start playing with that. And so this happens to be a Bill Clinton neuron. And so then they can do fancy things like look at different lures. So not the exact same target, but another picture of Bill Clinton, a foil, an unmentionable foil. This is the target. And then there are lures that are very similar, but there are also lures that are very different. And one of the cool things she found, and these are different cells for different people. This is an Anne Hathaway cell, this is the Clinton cell, and this is a Jay Leno cell. The lures can be the same person, but with different features, and some of the lures are very different, some are very similar. And then, of course, there are these foils of other people who are thrown in there, like here's Letterman next to Leno. And what she found, most interestingly, was that high performers showed a greater segregation between the targets and the lures. So this would be sort of evidence of pattern separation, which is something that we also see in rodents. So that if you're a good memory performer, you responded again in these, particularly in the CA3DG and subiculum, same regions we've been talking about all along, by the way. So that the high memory performance is associated with more target selectivity and more hippocampal activity to the target and less to the lures. But of course, you always have to ask the question, do we really have Jay Leno specific cells? And do we really have cells that are specific for Jay Leno in this tuxedo as opposed to a different tuxedo? Like, no, right? That would make no sense at all. The activity just has to do with the learning episode in time. We happen to have an electrode in, and we happen to get a neuron that responded to whatever stimulus that happened to be beginning at that time, which in this case was Jay Leno. And so it's really just that Leno happens to be, this neuron happens to be active when Leno appears, and that becomes incorporated into this experience of binding this stimulus with this event. Like today, I'm sitting in the hospital and getting electrodes in my brain. The activity has to do with a learning episode in real time, the sensory input of Leno with that temporal stamp, so to speak. And this is consistent with also the concept of remapping, which is found in rodents as well as humans, that those neurons could be Leno one day, but they could be somebody else or something else another day. And so it suggests also that place cells should also reflect the episode. Not that this cell is a place cell for this location, but it is this location at this point in time when I happen to be measuring it. So just to sort of conclude in broad ways about the functional anatomy that we can find in the human that I think is very similar to what we've seen in the rodents. Pre-entorhinal cortex, we see a lot of content-specific activation. So when we see places, we see place cell activity in perihippocampal place cell faces, activity in the face region. Everything goes through the entorhinal cortex. And then in this part of the circuit, where we have these recurrent collaterals, these reverberating circuits, this is where we see novelty, new association formations, consistent with these general encoding processes, putting new things together that didn't normally go together. And of course, the anatomy of this seems to be optimized to encode for this. Whereas in this part of the circuit, we've consistently seen activity associated with the retrieval process. So to conclude thus far, what I would suggest is then, at least in the human, the hippocampal processes content indiscriminately, but it processes the approach, the process, encoding versus retrieval in a highly specific way. And there are a lot of other processes that, of course, I don't have time to talk about that are being studied in humans as well as animals. I just will mention pattern separation, partial cue reconstruction, remapping, item versus source memory. There are a lot of other processes that take place. It's not just so simple as encoding and retrieval, but since we had a lot of data on it, it seems a good place to start. But within the hippocampus proper, it's the same functional circuitry regardless of the content of the task, regardless of what kind of information is being processed. So what I would suggest then is that the difference between the animal and the human hippocampus is not in how the hippocampus works, but really just in the unique information that's being fed into it. And humans get fed different kind of information like language and faces and consciousness. But we're doing the exact same thing with our hippocampus. And so in just a couple of minutes, I just want to touch on this second point, which is why is the hippocampus so vulnerable? So many things affect it. Viral encephalitis, anoxia, epilepsy is very prominent in the hippocampus and other parts of the medial temporal lobe, and stress and aging as well. The hippocampus is very vulnerable to stress. Stress, as we all know, impacts hippocampal structure and function. The hippocampus will actually shrink over time when exposed to a lot of stress. But I would say there's also a plus to the hippocampus being so responsive to stress because it's very important for learned fear responses, and these are essential to our survival. There are dense serotonin receptors in the hippocampus, and they respond to stress, and they do so throughout the lifespan. But it turns out that the density of those serotonin receptors are associated with superior memory. And this is a PET study by Vladimir Kepi, our colleague, with Dr. Gary Small, looking at this PET ligand, which is a 5-HT1A receptor ligand, very, very densely expressed in the hippocampus. And this is the correlation with memory. And this, not so many data points, but this is a correlation of 0.9. You just don't see correlations like this. That's a really high correlation with memory. And so it does suggest that there is an important component to this sort of stress responsivity and the ability of the hippocampus to process memory. And the other thing I want to mention is the horrible aging problem that we all have and aging risk. We've been studying the ApoE4 allele, which is a major, major risk factor for AD. And this is my colleague, Alison Berggren, who's been doing most of this work, looking at cognitively normal older folks that carry this gene. And one of the things that she has been showing is that even if you look at people in their early 60s who are cognitively normal, who, if they have the risk gene, they have shrinkage of the, basically thinning of the cortical ribbon in some of these key areas that are associated with Alzheimer's disease, like entorhinal cortex and subiculum. But functionally, want to show you some work by my graduate student, Tessa Harrison. There we are at her patient proposal meeting, showing some unique connectivity in ApoE4 carriers. So she got a bunch of young people from this Duke genetic study. So these are 570 people, 18 to 22. So they don't have pre-Alzheimer's disease. These are totally normal volunteers. And she did some analysis looking at graph theory network analysis. I'm not going to go into details. There are many ways in which the two groups differ, and many of them I think you might think would be, not very good for memory. But there is one thing that is. There we are again. I have like 4 of these, I swear. And we never did it on purpose, I promise. We even have matching two-tone candles. Crazy. These are 2 very important networks. The default mode network contains the hippocampus in its network, and this is the task positive network. And these are networks that are supposed to work in opposition to one another. So they should be very well segregated in order to be able to perform memory well. And what she showed was that in the young people, there was in fact greater segregation between these networks in ApoE4 carriers, suggesting that the ApoE4, while very bad when you're much older in life, earlier in life may confer some kind of advantage to memory processing. And this has been shown in other studies as well. This is a study showing ApoE polymorphism. ApoE4 is associated with improved attention and distinct neural signatures. And there's been something similar to this in the animal. This is sort of the animal version of ApoE4, showing altered synaptic plasticity and fear learning in those carrying the animal equivalent of a risk gene, where the memory and fear learning was actually improved in those animals. So, are there benefits of a vulnerable hippocampus? It's vulnerable to hypoxia, but on the other hand, there is a high baseline activity in hippocampus. It has high oxygen utilization, and that's very good for being able to encode information all the time. But if you've got that much metabolism going on, it's going to be vulnerable to stress. Epilepsy. Well, you probably all know that kindling is like a major model of learning. The hippocampus is very, very good at kindling. That's where everybody goes to study LTP. So it remembers these new firing patterns. That's a good thing. It's just a bad thing if you get the wrong firing pattern in there. It's just a nasty side effect. Sensitivity to stress. Well, the hippocampus facilitates memory for emotionally salient events that can increase survivability. That's a really good thing. But of course, it still means that the hippocampus has to be very sensitive to stress that comes in. And aging and genetic risk, it's possible that ApoE4 confers some kind of perhaps not fully known advantages in information processing. That is something that we now refer to as antagonistic pleiotropy. So in a way, we could think of sensitivity to input as really being the essence of what learning is all about. And that kind of sensitivity, unfortunately, necessitates this kind of vulnerability. So in broad conclusions, using high-resolution fMRI with similar tasking methods, we see heterogeneity in hippocampal subregions, just as we see in the rodent. Across content areas, we see the same processes, encoding and retrieval here, that have the same regional neuroanatomy. So the human fMRI data suggests that hippocampal function is process-determined, but content promiscuous. And this permits this tremendous plasticity and response flexibility to learning in any kind of situation. However, I'd argue that this very same feature of input flexibility creates an inevitable vulnerability in the hippocampus. And therefore, what is unique about the human hippocampus is not how it functions. It's really the same as it functions in rodents and other animals. But it's rather different in the content that it receives, including things like self-awareness and self-consciousness and language, these declarative aspects of memory. And so with that, I will thank you, and so will the yaks. Thank you.
Okay, I'd like to thank Susan and remind everyone that there's going to be a reception in the Gonda, first floor Gonda after a few questions. You willing to ask a few questions? So, is there some questions?
Oh, that's beautiful! And every time I see those unrolled hippocampi, they're just gorgeous. I'm thinking about the first task and the declarative memory of a wife who's not an unfamiliar person and has emotional content. And then I'm thinking about the fact that you were looking at homogeneity along this longitudinal axis. And when we think about the rodent hippocampus along that axis, which is, bent at a 90-degree angle, we kind of separate the dorsal hippocampus into more spatially encoded tasks and the ventral into emotionally encoded tasks. And I was wondering if you thought about that, or if there's any indication that human hippocampi might be.
Yeah, so we haven't studied that, but and I didn't have time to go into it, but different parts of the entorhinal cortex project to different parts along the long axis of the hippocampus. And so that is the reason for that dissociation. So, of course, we'd hypothesize that if we put in, for example, emotional faces or things that have that are getting input from different parts, that we know to be getting input from different parts of the entorhinal cortex, we should see the same association with humans. We just haven't done it yet. Don't know anybody who has. Ivanka.
Thank you for an excellent talk. I have a question about stress. Stress is interesting because animal studies shows a lot of damage of stress. However, there are a number of human studies that fail to do that. For instance, many PTSD studies where you would expect it. Now, the question is, are you aware if there is a special field if you in the parcellated hippocampus in humans that would be specifically stress sensitive?
So I think that's the same question that Anne was asking in a sense. So are there subregions of the hippocampus that appear to be stress sensitive? Okay, I have not studied it, but I believe that Nancy Psychot has. And I believe, and this is like an old memories, and I have retrieval deficits. So I believe she found it much more specific to the CA1, but I don't know I don't know that for sure. I have to go back and look at the paper. Yes.
Another short question: sex differences. Always coming back to that. Do you know if the fusiform, not the fus, yeah, it was not a fusiform, the one that is subiculum. No, fusiform. No, it's the fusiform. It's more because it reflects the visual activation. So would that be different in female compared to male? Again,
we have never done any gender comparisons in these studies. So I guess your hypothesis is that the visual information processing might be different for males and females, and therefore the hippocampal functioning might be different. Oh, that's a good idea. I've not looked at that yet. You could look at that.
Thanks. Wonderful talk, Susan. I have just two questions, I guess fairly broad questions about the dissociation between encoding and retrieval in hippocampus. So one is that retrieval, the act of sort of recalling a memory, is known to sort of strengthen the memory trace. And so there's this inherent interplay where retrieval is one of the best ways to learn information.
Re-encoding.
And the second is sort of how your thoughts on how the hippocampus knows when it encounters information in the world, whether to be doing sort of encoding and retrieval. Is it just a function of novelty, or does the hippocampus actually sort of switch into a back and forth between an encoding mode and a retrieval mode under top-down control?
Oh no,
just sort of wondering.
Yeah, I don't at all believe that there's top-down control over that. I think that it just responds to what is new, and if it sees something before, it's an automatic process that recapitulates that loop. And if you'll notice in the subiculum graphs, there was activity for retrieval that was greater, but there was still encoding activity. And I think that has to do with that re-encoding process that you're talking about. But I don't believe in the top-down control theory. I think that would be equivalent to the camel in the yak thing. I think that the hippocampus sees something new. Even if it doesn't see something new, it sees something old in this context, it's still going to be encoding. Okay. Dr. Fuster.
Susan, great lecture. I'm very proud of you indeed. I have a comment with respect to the apparent discrepancy between the rodents' data and the human data. Mm-hmm. You have covered that or resolved that discrepancy in part. I think that the answer to it lies in phylogeny. In lower animals, such as rats and mice, navigation touch, and olfaction play a very vital role. And these are functions of the limbic system, and in particular, the hippocampus. The hippocampus is cortex, after all. So what happens with phylogeny is that those very important survival functions are farmed out to the neocortex, to the new cortex. But what is retained in the hippocampus is the ability to encode and retrieve information of all kinds, including, of course, those.
Yes.
So that what we have is a neocortex which is specialized in learning and returning information of high complexity using perhaps the same mechanisms of association that prevail in lower species in structures like the hippocampus.
I completely agree.
Again, congratulations and thank you.
Thank you. Thanks so much.
Okay, I think with that we can go to the first floor gondola room and have some lunch. So again, thank you very much.
Thank you.
Jolly good.